Okay. So this is the outline of my presentation today. I have 5 bullet points, so to say. So briefly, I want to review all these 5 points, and then I will go through that later on. So the first one, the great majority of our knowledge on how the human brain functions seems to be based on correlation studies. Correlation studies are all those studies where we analyze how the brain or some part of the brain reacts to the presentation of some stimulus or during some cognitive or verbal linguistic task. So belong to this set a wide range of techniques, including single unit recording, scalp EEG, intracranial EEG, MEG, and all brain imaging studies, including, of course, fMRI. The second point is that all correlation studies suggested up to now 3 main models to map the relationship between structure and function. So the first and the more classic one is the so-called one-to-one mapping. For example, fear is encoded by the amygdala, and amygdala encodes fear. The second one is the one-to-many or many-to-many relationship. Briefly, the point is that, for example, the amygdala encodes fear but many other things, and it depends also from the link that amygdala established with some network or some other network during some specific in some specific moment, and belong to this category a long range of hypotheses close to my house, so to say. For example, in the neural exploitation hypothesis, the shared circuit model, neurocycle, and of course the neural used by Anderson. And the third model is what we can call psychological constructionism. Yesterday we had Basel talking about this. Basically, according to constructionism, there is no clear relationship between structure and function. So, for example, fear can be driven by amygdala activation, but not necessarily driven by amygdala activation. And at the same time, amygdala can be active without producing fear, but doing something else. So it seems that many current debates seems to have come to a standstill. The core part of my talk is that, in my opinion, it could be that all correlation studies suffer from the same epistemological problems described by Quine in the radical translation experiment. And if I'm correct, it means that the choice between different explanations available is not a matter of increase the number of evidence. So it's not a matter of increase the number of experiments, but something different. And the very last point is that maybe interference studies can help to escape this impasse. Interference studies are, okay, we can consider all techniques in cognitive neuroscience belonging to one of the 2 sets, correlation studies or interference studies. In correlation studies, the experimenter investigates how a part of the brain reacts to the presentation of some stimuli or some cognitive task. During interference study, the experimenter evaluates how the subject, and not the part of the brain, how the subject reacts during the activation or deactivation of some specific part of the brain. So in the second set, we have electrical stimulation, chemical stimulation, magnetic stimulation, brain lesion, and so on. Of course, we can have very different temporal and spatial resolution in both correlation and interference studies, but the main characterization, I mean, is this now.
Yes. No?
See. So, as I said, correlation studies suggest different types of models to map the relationship between structure and function, and we can group all the proposals into big main categories. On one end, we have essentialist approach. We can consider one-to-one and one-to-many or many-to-many mapping being part of this category. And on the other end, we have psychological constructionism. And they are in competition one to the other, basically, if you look at the literature today. And we can find these 2 approaches in a large range of topics in cognitive neuroscience. For example, the very well known is the field of affective neuroscience, where it is clear where people in the field of essentialist tradition think that, for example, we can find some kind of relationships from some structure and some function while people in the constructionist story think this is not possible. But the same approach is evident also in motor behavior studies. For example, people in the tool story, in the tool behavior story, is asking whether the inferior parietal lobe contain manipulatory knowledge or motor program, essentialist version, or mechanical reasoning, which is part of the constructionist story. And the same is also true in the visual perception studies. For example, does the inferotemporal neuron encode object identity, essentialism, or vice versa, some kind of primitive component, which is part of the constructionist story? So my point, thank you, is that correlation studies seems to suffer the same epistemological problem of the kind described by Quine in the radical translation. I think that all of you probably know very well the original experiment by Quine, so I think that a brief, even because time is short, a brief summary can be sufficient. So basically, Quine described the case of a linguist having to translate the language of a tribe that has never had contact with English speaker, and the only help is from a native. So basically, the linguist and the native work together, and from time to time, then the natives say, pointed at something, this is that, or this is this, this is that, and so on. So, and observing, and of course, according to this ostensive approach, the linguist will translate, for example, this with chair. So observing the scarring of a rabbit, the native produced the famous statement, gavagai, and the linguist translate gavagai using rabbit, because they both see the rabbit. But the point, say Quine, is that in principle, we can even translate gavagai with undetached part of rabbit or rabbithood. And the very big problem is that each time we see a rabbit, we also see undetached part of rabbit and exemplification of rabbithood. So all the alternatives are perfectly plausible, and increasing the amount of empirical evidence cannot be of any help in disentangling between the two. And what also Quine suggests is that starting from this alternative, we can produce different manuals for translating one language into another, one containing rabbit and the other one containing undetached part of rabbit, and we can, both the manuals are consistent, but we cannot move from one manual to the other. So that's in brief, the point by Quine. So my point is, what if essentialist approaches are providing us with one manual and constructionism is providing us with another manual, and we cannot just increase the number of evidence to see whether the first is correct or the second is correct? No, that's the main point. And see? So my hypothesis is that standard procedure in place during correlation studies, and I repeat, correlation studies include single unit, skull PG, MEG, brain imaging study, which is something like the 80 or maybe 90% of our knowledge on the human brain function. What if all these studies belong, I mean, the structure of all these studies is similar to the one described by Quine? Please. And so, for example, let's consider this kind of translation. The linguist in this Quine radical translation can be replaced by the brain researcher. The native, he's the brain, or in particular, one neuron if you're working with single neuron recording, or brain network if you're working with fMRI, multivoxel pattern analysis, for example. And the scarring of a rabbit is the experimental stimulus, even because we can imagine to modify the original story by Quine. So let's imagine the linguist, instead of working together with a native, just present the native with a rabbit. It's exactly the same as the original. So he presents the rabbit, and the native say gavagai, then hide the rabbit, then present a new rabbit, and you say gavagai, and so on and so on. Now, just look at this video. This is an original movie from Hubel and Diesel, original experiment in primary visual cortex, where they discovered that the primary visual cortex the plot simple cell. The, stop nothing. Okay, so they are mapping the visual field of one neuron, and they find that this is the optimal stimulus to trigger the activity. The sound, the noise, is the increase on firing rate of the neuron. Now the light is outside the visual field, the receptive field of the neuron, which is now marked by these crosses. So this is not a rabbit anymore. Now the rabbit, guy, and this is maybe an elephant or whatever you want. But as soon as the light crosses the crosses, we hear Gavagai again. Gavagai, okay? And this is another animal. No rabbit here. Okay. Tracy. So, the linguist, no, sorry, ancora. The linguist is the brain researcher, the native, no, dietro. He's the brain, the scarring of a rabbit is the experimental stimulus, and Gavagai is spiking activity if you're recording single neuron, increase of bold activity, or strength in correlation according to the technique you're using, or evoked ERP, evoked related potential. So whatever, I mean, the way you measure brain activity, we are always in the same original situation described by Quine. Okay. A case study. This is an original paper by Tanaka and co-worker back in 1991. They were recording single neuron from the intertemporal cortex, and they found some neuron firing during the observation of a tiger. They were recording from a monkey, a macaque. And they were presented the macaque with tigers, and they found some neuron firing during the observation of the tiger head. So one could argue that this neuron encode or represent, if you want, tiger's head. However, they also present the monkey with black and white tigers, and the neuron was still firing. Then they presented the monkey with some degraded version of the original picture, such as the third, the fourth, and the last one, and the neuron was again firing. At the end of their work, they tried to present the monkey with white square by their own or black square by their own, and the neuron was not firing anymore. Okay, so what we can argue, we can say that, for example, that the ontology of this neuron does not include colored tiger's head, but rather black and white overlapping rectangles, which is correct. However, can we say that we identify the true selectivity of this neuron? I think not, because, for example, from a constructionist point of view, which is the one supported by Tanaka himself, but also, for example, by Pierre Jacob and Marc Generot, the analysis of visual stimuli affected by intertemporal neurons is not complex enough to specify a particular biological object on the basis of a single cell discharge. However, from an evolutionary point of view, we can even argue that this neuron encodes trigger-like patterns plus false positive for evolutionary purposes, because it makes a lot of sense for the monkey to see tiger where there are no tigers, for example. Okay. Another example, essentialism versus psychological constructionism in the field of affective neuroscience. So we can imagine essentialist supporters of essentialism say that there are some relationship between specific emotional state and brain activation. However, the psychological constructionists reply, meta-analysis show that there are not one-to-one relationship between emotion and brain region. But what we can even reply, however, there are one-to-many relationship between emotion and brain region. And at this point, which is a very nice thinking, a pattern that successfully distinguishes the member of one category from the member of another is not a fingerprint, in a sense. It is an abstract statistical summary of a variable population of instances. And again, essentialism, if we were to take this logic to the extreme consequences, we would come to the paradoxical conclusion of having to abandon concepts like vision, speech, hearing, and so on, and so on, and so on. So, and so on. No, so the point is, The situation in cognitive neuroscience is even worse as compared to the original presentation by Quine, because Quine say, okay, we are not sure that we cannot say that rabbit is the true translation of GABA guy, but it makes a lot of sense because all of us, we share the same ontology, if you want, because we are all person, and so it makes a lot of sense. We can apply some normative principle and to decide that this is meaningful, even if it's not logically the only possibility. Okay, this option is not even possible in the field of neuroscience because we don't share the same ontology of a neuron. It doesn't mean, it has not any sense. It's meaningless. Okay, so my point is that maybe we can try to include in our knowledge of the brain also information from interference studies, because during interference studies, such as, for example, electrical stimulation, chemical stimulation, and so on, we are not hypothesizing what the neuron refers to, but we are just judging the behavior of another individual after the activation or deactivation of some specific part of the brain, okay? So, for example, let's consider, and this is my last slide, the next one. Let's consider, for example, this has been retrieved from Lindquist's meta-analysis, and she compared essentialist approaches to the brain versus psychological constructionism. And what she say is that, for example, many people say that the anterior cingulate cortex is involved or encode happiness, but according to a constructionist view, the anterior cingulate cortex encode core affects. And the same is true for the insula. The anterior insula, according to many people, encode disgust, while some other people say core affect again. All these data are meta-analysis from correlative studies, brain imaging studies. Now, and this is my last, I want to show you, okay, if it works, what happens when we stimulate.
Sorry, no, we have to open it manually, sorry. Sorry, Fausto.
When we, stimulate the anterior cingulate, this is a patient with an electrode in the anterior cingulate cortex, and at a certain point, the surgeon will stimulate, and you will see what will happen, even if it's already clear because it's in the title. Stimulation now. While this is what happens when we stimulate the insula. Okay, so this is another perspective to investigate the brain. Finished.